Welcome back to this Great Academy lecture in biology where we're beginning enzymes. Now we consider them to be the controller proteins of life, and we have to know a lot about them. First of all, the definition is critical. You have to know that they are biological catalysts, and that means they speed up or slow down a reaction, but they themselves are not used up. But it's also a really good idea to say they are protein catalysts, because then when you have that in your definition, when they ask you what the nature of it is, you're safe because you know it's made of protein and is in there. They control all the reactions inside an organism, all of the reactions, like the metabolism, the sum of all the reactions are all controlled by enzymes. So photosynthesis, respiration, protein synthesis, making proteins, all of those reactions are made and controlled by these guys. So it's important that we know they exist in the cytoplasm and they are actually majorly important. We consider them to be the controllers of life. So we have to know a number of things about them. First of all, as I said, they're made of the biomolecule protein. And from the food section, you should remember that means that they're made of the elements carbon, hydrogen, oxygen, and nitrogen. And from the cells, from the organelles, you should know that the ribosomes build proteins, so therefore they build enzymes. So there's the link. They're three-dimensional in shape, like all proteins are. Now, when we come to the word specific, that's really, really important. They work on particular substrates. That's what they work on. An enzyme works on a substrate. And they have optimal, now I call that Goldilocks, just right. They have optimal temperatures and they have optimal pHs. In other words, they function at an ideal temperature and an ideal pH. It's acidic or basic, depending on the type. And they are affected, not effected, that's a result. They are affected by changes in the pH, the temperature, and the particular substrate concentration. So if they're in too much, they won't be able to work as fast. But they are extremely fast workers indeed. Now, the last thing is still relating to the fact that they're protein is that extreme changes in temperature, particularly, and in pH, and even something like whipping it, breaking it up, that will mean that the enzyme becomes denatured. Now, I always want people to think about an egg. And when you open an egg, you can see the transparent part of the egg, the liquid, the white. We call it the white. We call it the white of the egg because when we put it on a frying pan, the protein in the white of the egg, which was transparent, turns white. It becomes denatured. You can't turn that back the other way. You can't reverse that job. And so that's what denatured is. And these enzymes, when they get to a particular temperature, they're beyond their acceptable temperature, then they are actually going to get denatured. And when they denature, they lose their three-dimensional shape and they fail to function. Okay, so there's 7 things that you have to know. And the remainder of this chapter and this section is that they actually want you to show that these are things that actually happen. Now, how do enzymes work and what do they work on? They're not rigid. They actually take up the shape. They're a bit like the bean bag. When you sit into it, it takes up the shape of the substrate. Now, what they're working on is a substrate. They're always working on a substrate, and then they make a product. And this is what you call induced fit model. Okay, so here are the words specific again. Now, when you hear the word specific, the word that should come out in your answer should be the opposite, the same word, except it's a different word. It's particular. So the substrate specific, oh, I'm getting bad now, is the enzymes will only work on a particular substrate. Particular substrate. So what I've done here are all the enzymes that you have to know. And what I've done is I've made it really look like it's cut and paste deliberately. So lipase works on lipids. All the enzymes have A's in them. Protease works on the substrate protein. Maltase works on the substrate maltose. Sucrase works on sucrose. Amylase works on starch, and the old-fashioned name was amylose, or the technical name for it, but we still call it starch. And of course, there has to be always an odd one. Catalase, which is the fastest working enzyme, it works on hydrogen peroxide, and we will deal with that later in the lesson. So we're going to introduce you to good old amylase, and so I've made him look a little bit like a purple or lilac blob, and I've just given him A. And he's going to literally let you know what he's done. It's just in a speech bubble. This is his active site. This is where it works. He's a metabolic protein, so therefore he is going to adjust, and he's part of the metabolism. And he likes temperatures just and above and below 37. That's its optimum temperature. And it will not work unless it's in the optimum pH of 7, 7.2, but certainly not acidic conditions. Okay, now if he's specific, what does he work on? He only works on starch. So what have I done? I've drawn a little line of starch. Glucose is all in a row, which is starch. And here is a big long strand of starch, maybe from a chip or a piece of bread or a pasta or something like that. And this amylase in your mouth starts to get to work on it. And you notice the way it actually has now slotted in nicely against the hexagons of 2 glucoses in the starch. And what he does is he goes 1, 2, cut, 1, 2, cut, 1, 2, cut. Now you don't need an awful lot of them. These 2 enzymes here, these 2 enzyme molecules, and there's a third one. And look at all that starch. And for those 2 boyos, they have broken it down into a disaccharide. That is a polysaccharide, and that's a disaccharide. And the disaccharide with 2 glucoses is called maltose. It's also now the product molecule. They're disaccharides. And the beauty is he's free to do the whole thing again and again and again and again and again. And that's what's the beauty about enzymes. Now, the other enzyme that you have to deal with in terms of little practicals is an enzyme called catalase. So what I did was I took something lilac again, but this time I tried to make it look like a stretched out beanbag. It's just distorted. And the enzyme is catalase, and this is its active site, this wobbly area here. Again, it's a 3-dimensional protein. And this time it's going to work on a chemical that you have to know. Biology students do have to know some chemicals, not too many, but this one is really important. It's called hydrogen peroxide, and it's actually like water with an extra O. You all know what the formula for water is, H2O. But when you've got an extra O, it's H2O2, H2O2. And I've drawn it here like a little single bonds with 2 oxygens here. Now, what I have to draw is I have to draw 2 of them. So here's a hydrogen peroxide molecule and another one. This is not your friend. This would give you a sting on your skin. If you got it on your skin, it would sting like a nettle, but it wouldn't go away too quickly either, and it would cause your skin to go white. It is made in metabolism in your body, and the fastest enzyme in the world is present in every living cell. This guy is present because the moment that is present, it will disintegrate the cell. It'll cause cell death. So this guy has to break it up the millisecond that it's made. And that's why it is the fastest known enzyme. And you can get this from anything. And in the case of when we're doing it, we actually chop up some celery and get it out of. You could have used liver from the butcher or anything really. Blood will do the same job. But we're doing it with celery, thank goodness. But anyway, this is the hydrogen peroxide, 2 molecules. Watch. Can you see that the 2 of them are now sitting down in the active site? And while my skills in computer graphics don't show it, it's like as if this now closes in on it. So the active site adjusts slightly to fit the H2O2 molecule. You see this way, like water, except it's got an extra molecule. Now, they sort of like jiggle it like a Rubik's Cube, and then boom, out it pops. And what do you pop out? Oh, look, H2O, H2O, and oxygen. I'm going to move that down. I'm going to sort of let you see. Can you see oxygen? O2 is released, and water. Now, oxygen is a gas, so it's going to bubble out of whatever it's in, and the water is going to just stay where it was. Now, we're going to see that in action. So the source of the catalase enzyme is the celery. In other words, the celery isn't the enzyme itself. The enzyme is inside the cells of the celery. So celery is described as a source. The second thing is the substrate. What is the catalase going to work on? Well, it's going to work on that hydrogen peroxide stuff. That's the catalase is going to work on hydrogen peroxide, and you do need to know that name. Now, if you wanted to catch any gas, the best way to do that is to put washing up liquids in to the mixture, and the bubbles of oxygen will then make it get trapped and form a foam. And then these kind of substances are going to actually keep the solution at a particular pH. So if you want it at 7, it's going to stay at 7 when you add a little buffer liquid. And then the water baths, well, you saw the water bath when we were doing when we talked about the food test, the Benedict solution test for reducing sugars. It is just a water bath. It's just going to keep the temperature constant. Or you'd have 2 or 3 water baths and you are going to vary the temperatures. Okay. They're what you need. So our first mandatory practical involved varying the pH so that we can investigate how effective catalase enzyme is at different pHs. Now, since this course started in 2002, they have never asked you results for this because it doesn't quite work as you'd like. But they've often asked you to explain what's happening. So let's have a look. Get out of the way, Stephanie, and let's have a look. So what is my picture showing you? Well, first of all, you have a water bath, and the water bath is set at 25 degrees. So that's constant. I'm keeping that the same. I am not varying that. And look, I have chopped celery in 5 graduated cylinders. And I have hydrogen peroxide sitting in small graduated cylinders, also coming to the same temperature. So this is at the start. Now, the other thing is, let's have a look at what they've got in common. There's 10 centimeters cubed of buffer. There's 5 grams of chopped celery into each of these. There's 3 drops of washing up liquid into each of these. And they're all sitting in a water bath set at 25 degrees, which is the optimum temperature for plant activity, plant cells activity. But what have I changed? I have changed the pH. So while I have put in a buffer here, it's a buffer of 4 that makes it quite acidic in here. I've added a buffer of 7 here, 8 here, 10 and 14. Generally in a lab, you wouldn't do that one, but it's just to let you see what you have. So they're all ready. And just when you want this to start, they can all be individually done. You don't have to do them all at once. It's just my pictures trying to show you them all at once. And so you start your timer as you add the hydrogen peroxide in. And as you know, what's going to happen is that's going to react with the celery, the catalase in the celery, the chopped celery. You're going to have the oxygen made, and hence you're going to see foam. So we're ready. And boom. After 1 minute, you measure the height of the foam. It didn't look like very much happened there. This certainly looks like it was quite vigorously making oxygen. This is even better, because the height of the foam really is a reflection of how much activity was going on. And that means the catalase enzyme has been working most efficiently in these 2 and much less efficiently down here. So something's wrong. The temperature was right and everything else was kept constant, so it must have been the pH. So if you take these measurements of the heights of the foam or the height of the volume and translate that into a graph. Now, this first one, take this one with a pinch of salt. Look here, 4, pH of 7. Now, I only did this because I want you to see where the dots appear in a graph. 8, 10 and 14. But as you know, this should be evenly spaced. So it should be 4 and near at 7, 8 should be nearly there, 10 should be there, and 14 should be over there. So this is a more correct image. So can you see this up and down? You want it to be sharper than this picture, but I wanted you to see how those values were got. But in fact, 4, 7, 8, 9, 10, and 14 is over there. So you should see a bit of a spike up there. That is critical. Now you can see that this area here is the optimum pH for the enzyme catalase. Now, they used to think that it was 9, but it's, it works just as well with 7, 8 or 9, really, 8, 8.5. But certainly it doesn't work with 4, and it certainly doesn't work with 14. Now, You've done that. You come back into the next day and you this time want to see how the catalase enzyme varies with different temperatures. Okay, so you're going to vary the temperature. Let's have a look at this photograph and pause this video to have a look at it yourself for a minute. So here we have 4 different water baths, and as you can see, I've put the temperatures 10, 20, 30, 40. I have the hydrogen peroxides ready, and look what I've got, 5 grams in all, 3 drops of washing up liquid, 9, the buffer of pH 9, could have used 8, but it was 9 that they recommended in the Department of Education, and there was 10 mils of it in each one, so nothing varied, the same apparatus, the only thing that varied was the temperature. And when you started your stopwatch and you timed 1 minute for the foam to be produced, what sort of results would you expect? Well, what you'd expect is that a good temperature would work. And now let's have a look. That one seems to have done okay, but it's 10 degrees. Remember, enzymes, you try texting in the cold when you're cold and your fingers are cold because the enzymes in your fingers aren't able to work because they're not at their optimum temperature. This one looks okay, 10, 20 degrees. 30 degrees looks really good. 40 is pretty much the same, a little bit lower. Convert that to a graph and you get this classic bell. If I had gone to 50, that would have started to take a dramatic nosedive because after that temperature, it's gone past a temperature where the proteins in a catalase enzyme would be denatured and they would totally fail to function, no foam. Now, so here we come to the third practical. This is the mandatory practical where we want to see the effect of heat. Now, you could have done this experiment and done it at 50 and 60 and 70, and you would have seen a sharp drop. Why isn't it sharp down there? It just means that the enzyme isn't fully denatured, but it's slowed down. Okay, now, what about this picture? Oh, they're both at 25 degrees. It looks like I didn't change the temperature, but look closely. This is boiled celery. So we took that celery and we boiled it for a while. This celery was raw celery, didn't get boiled. You still sit them both, let that cool, of course. And so the celery is still celery, but all the little enzymes in there have been cooked, have been denatured. And you have 5 grams of celery in both cases, and the buffer is still pH 9 and 25 degrees. So there's constant temperature and all the rest. Do remember that a water bath is thermostatically set to stay at 25. A thermometer doesn't, that's a classic mistake. A thermometer doesn't actually keep it warm. It just tells you the temperature. It's a classic mistake. Now, what about that? Before and after. This foam is just from the result of the washing up liquid. It really won't make any foam even after 2 minutes or 3 minutes. This, on the other hand, will foam, and it will continue to foam until it runs out of the hydrogen peroxide that you've added. So that is what you would do. Now, some years ago, a question came out and it really threw students, and it was never asked before, and in the marking scheme they just say any reasonable attempt. But the question asked you, how would this experiment look in a graph? Now you say, well, you'd understand how to do the other ones, but this one you've only 2 points. The first moment when the dot was here and the second moment when it was there or there. And I remember setting it for my own students, and they were finding it difficult, except one student said, well, what if you had waited for a minute and another minute took a reading a minute later and another minute? What you would see is, okay, no foam. After 1 minute, you had that much. After the second minute, you'd still have that much. And the third minute, it would never go up. Whereas this would go up, but then it would stop because it would run out of hydrogen peroxide anyway. Okay. So that's the first part of enzymes and the 3 mandatory practicals that are associated with it. In the next part of this lesson, which is in a different, shorter time, we're going to look at capturing some of those very useful enzymes and putting them to work for us. Not that we don't already do that in our cells and in our bodies, but taking them out of where they are and making them work for us in industrial biochemistry and biological action in bioreactors, well, that's next lesson. So thank you for watching this great Academy lecture. Until next time, happy learning.